Good day, everyone. So I am here to discuss to you the structure and functions of your cell membrane. So, if we're going back to our discussion in week 5 lesson in midterm, so we have discussed the different cell parts and functions, which includes your cell membrane as one of the three fundamental parts of your cell. And this cell membrane plays a very important role in terms of the total function of the cell. So first, what is your notion when you hear the word cholesterol? So sometimes when we encounter the word cholesterol, we usually perceive this word in a very negative way, since what we usually hear when we hear the word cholesterol causes cardiovascular diseases. But then again, not all the times this cholesterol has a negative impact. So it is in the person who have the control in their intake of foods that are high in cholesterol. As they say, too much is not good. So everything should come in moderation. And when we say moderation, we should know our limits. So what is this cholesterol? So you must probably know that this lipid class can cause cardiovascular diseases. Yet we still enjoy various cholesterol-rich foods such as your fries, fatty beef patties, eggs, and cheese. So this LDL cholesterol, or the low-density lipoprotein cholesterol, which is also dubbed as the bad cholesterol, is particularly dangerous because it can accumulate in the arterial walls and can result in clogging. So normal cholesterol levels in the body should be less than 200 milligrams per deciliter in adults. So there are various types of the different cardiovascular diseases that this cholesterol can give. So one of which is your arteriosclerosis, which occurs when the blood vessel that carry oxygen and nutrients from the heart to the rest of the body become thick and stiff, so sometimes restricting blood flow to the organs and tissues. So these healthy arteries are flexible and elastic, but over the time, the walls in the arteries can harden, a condition called hardening of the arteries, and that is because of the buildup of this cholesterol. So one specific type of arteriosclerosis is your atherosclerosis. So arteriosclerosis is the buildup of fats, cholesterol, and other substances in and on the artery walls. So this buildup is called the plaque. The plaque can cause arteries to narrow, blocking blood flow. So the plaque can also burst, leading to a blood clot. So although arteriosclerosis is often considered a heart problem, it can affect arteries anywhere in the body. So arteriosclerosis can be treated. So one of the most effective way to treat this arteriosclerosis is a healthy lifestyle habits. So there are some manifestations of this mild arteriosclerosis which usually do not have any symptoms. But when an arteriosclerosis is, or what I say is, the symptoms for arteriosclerosis usually don't happen until an artery is so narrowed and clogged that it can't supply enough blood to organs and tissues. So sometimes a blood clot completely blocks blood flow. So the clot may break apart and can trigger a heart attack or a stroke. So some symptoms of moderate to severe arteriosclerosis depend on which arteries are affected. So for example, as what I mentioned, there are various symptoms of this moderate to severe arteriosclerosis depending on which arteries are affected. So if you have arteriosclerosis in your heart arteries, you may have chest pain or pressure, which you call the angina. Now, if you have arteriosclerosis in the arteries leading to your brain, you may have sudden numbness or weakness in your arms or legs, difficulty of speaking or slurred speech, temporary loss of your vision in one eye or drooping muscles in your face. So this signals as transient ischemic attack. Untreated transient ischemic attack can lead to a stroke. So if you have arteriosclerosis in the arteries in your arms and legs, you may have symptoms of peripheral artery disease, such as leg pain when walking or claudication, or decreased blood pressure in an affected limb. So if you have arteriosclerosis in the arteries leading to your kidneys, you may develop high blood pressure or kidney failure. So that's why most people sometimes have this high blood pressure or kidney failure, because the most affected organ usually is the kidney. Now, despite the harmful effects of cholesterol, it is an important structural component of the plasma membrane of the cell. So this lipid is still required by the body to perform its biological functions. Such functions include the synthesis of hormones, bile acids, and vitamin D. So in addition, cholesterol is also an important structural component of the plasma membrane of cells. So especially in the aspect of membrane fluidity. So you can imagine how a stiff membrane will be in the absence of cholesterol. And a stiff and rigid membrane then implies impairment of various biological functions. So note, however, that our bodies produce the cholesterol needed for plasma membrane structure. So this is not obtained from our diet. The plasma membrane, being the primary barrier of the cell, is very important because the impaired function may put an organism's life at risk. So for instance, a diabetes is an example of a defect in the cell's plasma membrane. So a diabetic's plasma membrane does not respond properly to insulin, so the hormone that lowers the blood sugar. Consequently, a diabetic individuals need insulin maintenance. Aside from regulating what goes into and out of the cell, the plasma membrane is also responsible for the communication of cells with one another. It is also responsible for the different chemical processes happening inside the cell because of some membrane-associated enzymes. So the membrane's efficiency in doing its role is very important. Now, what do you think is the relationship between the structure and function of the membrane, or how does it contribute to the cell being the fundamental unit of life? So we can recall from the discussion in our midterm week 5 that the plasma membrane is one of the fundamental cellular structures, aside from being known as to be the gatekeeper or the primary barrier of the cell. It plays other important functions because of its structural components. Now, as the primary barrier of the cell membrane prevents the cellular components from being mixed up with chemicals of the external environment. So it also receives information or stimulus from the environment to initiate necessary response. So lastly, the membrane is also flexible enough that it allows the cell to move and grow. So based on our discussion in our week 8 and 9 lesson in midterm, so we have discussed about mitosis and meiosis, and the very first phase that the cell undergoes is what you call the interphase. Specifically in G1, what happens there is that the cell grow and the organelles double. So why is it that this cell grow and that grow? It's because one of the organelles that is responsible for the cell to grow is the flexibility and the fluidity of this plasma membrane or this cell membrane. So without this flexibility and fluidity of the cell membrane, this cell will not grow during the cell division. So the cell membrane is one of the great multitaskers of biology. The purpose of the cell membrane is to hold the cell's different components together and protect it from the environment outside the cell. So the cell membrane also regulates what enters and exits the cell without losing too many nutrients or taking in too many ions. Remember that this cell membrane is selectively permeable as it regulates what goes in and out. So it does not allow all materials to enter or exit the cell. Now, once the cell membrane does not become selective, organelles will not receive enough materials from the outer part of the cell, affecting the organelles' function, thus affecting how living organisms live and function. So the cell membrane functions like this to secure the safety of the entire cell. If the cell membrane is not selectively permeable, foreign substances will invade the cell, so destroying its usual function. So a membrane in the cell's interface with the rest of the world, it's the gatekeeper, if you will. Now, this phospholipid bilayer determines what molecules can move into or out of the cell and is mainly responsible for maintaining the delicate homeostasis of each cell. So the cell needs to have a stable internal environment. So the temperature inside the cell should just be maintained to make sure that the function or the overall function of the cell will take place smoothly. Now, we have here the different models of the phospholipid bilayer that is proposed by some scientists. So the first widely accepted model of the plasma membrane structure was proposed in 2000 and James Danieli. So it was based on the railroad track appearance in early electron micrographs. So they theorized that the structure of the plasma membrane resembles a sandwich, with protein being analogous to the bread and lipids being equivalent to the filling. But then again, a new model was introduced. A better model was introduced and explains both the microscopic observations and the function of the plasma membrane. Well, it was proposed by Sir Jonathan Singer and Garth Nicholson in 1972. So the explanation proposed by Singer and Nicholson is called the fluid mosaic model. Why is it that this is called the fluid mosaic model? So the fluid mosaic model describes the plasma membrane structure as a mosaic of components that give the environment or that give the membrane a fluid character, including phospholipids, cholesterol, proteins, and carbohydrates. Now, we have here the general functions of your cell membrane. So one of which is your primary barrier of the cell. The second one, it receives information or stimulus. And number 3, it allows the cell to move and grow. So here is the structural components of your plasma membrane, which consists of a diversity of lipids, carbohydrates, and proteins. Now we have here the different structural components of plasma membrane, which includes your phospholipid bilayer, the molecules embedded in the membrane proteins, and carbohydrate chains. So first, we're going to discuss about the phospholipid bilayer. So based on the previous knowledge we have, the plasma membrane is mostly composed of phospholipids, which are composed of 2 distinct regions. So this nature of the plasma membrane was first proposed in 1925 by Dutch scientists Evert Gorter and Francois Grendel. So their proposal said that the phospholipid is an amphipathic molecule that has both a hydrophilic region, or the water-loving phosphate head, and a hydrophobic region, or the water-fearing hydrophobic tail. So this nature of the phospholipid molecules explains why they form a bilayer in water or an aqueous solution. Now, since the external and internal environment of the cell is mostly composed of water, the hydrophilic polar heads of the phospholipid molecules naturally associate with it, or it interacts with the water. So likewise, the hydrophobic or nonpolar tails associate with each other, or they are in between the two phospholipid heads. Now, a phospholipid layer alone will be troubled because of the presence of these two conflicting forces. So that is, one phase has natural affinity of water, while the other phase repels it. So the formation of two layers of phospholipids resolves this conflict. So it becomes energetically stable for the bilayer that the fatty acids face each other. So while the polar phosphate heads face the natural aqueous internal and external cellular environments, detergents such as soaps are also amphipathic in nature. So because of the similarity in nature, so high concentration of detergents can solubilize the cell membrane, thus disrupting them. So this explains in experiments required that this explains why in experiments that require the disruption of cell membranes and other internal membranes, such as the nuclear membrane or the nuclear envelope, amphipathic substances such as detergents are used. So the phospholipid can also move in the bilayer in several ways. So a phospholipid can move on its own axis through the rotation. So it can also move sideward through lateral diffusion, or it can also move from the inner layer to the outer layer or vice versa through the transversal diffusion. So the phospholipids do move in many ways. So these movements are essential to maintain their fluidity. Later on, we will discuss what is this fluidity. Why is it so important for the cell membrane to become or to have this what we call the fluidity? So phospholipids are constantly moving because stagnant phospholipids would hinder substances from entering and exiting without the cell. So imagine the cell membrane as a stiff wall which it does not move. So it's very hard for the materials that is synthesized inside the cell to go out the cell membrane. So it is very important that this phospholipid would move or will move in order for the synthesized materials to go out and outside the cell. Now, how is this plasma membrane described by the fluid mosaic model? So membranes are not rigid but rather fluid and flexible structures. So biologists use the fluid mosaic model to describe a membrane structure as composed of diverse protein molecules suspended in the fluid phospholipid bilayer. So this, as what we have mentioned, this model was proposed by Sir Jonathan Singer and Garth Nicholson in 1972. So the lipid content of the membrane is responsible for its fluidity, which prevents it from solidifying as external temperatures drop. So the presence of cholesterol molecules specifically helps maintain membrane fluidity, particularly by preventing the membrane from becoming too solid or too fluid at higher temperatures and too solid at lower temperatures. Now, the fluidity of the membrane is important for two reasons. So it imparts flexibility of to the membrane, which is important for cells that particularly moves, such as this amoeba, which is performing a cytoplasmic streaming. The second one, the fluidity allows the synthesized membrane proteins and phospholipids to be easily incorporated into the membrane. So there are synthesized membrane proteins and the phospholipids and carbohydrates that is processed inside our cell, specifically in the endoplasmic reticulum, the ribosome, the smooth endoplasmic reticulum, synthesizes these proteins, carbohydrates, and lipids, which is transported to the Golgi apparatus, and this Golgi apparatus will then transport these materials outside the cell membrane, and because of the fluidity of this phospholipid, those synthesized materials is incorporated in the cell membrane. Now, aside from the cholesterol embedded in the hydrophobic portion of the lipid bilayer, the fatty acids of the phospholipids themselves contribute to the fluidity of the membrane. So as shown in this figure, some of the phospholipids have unsaturated bonds, and that result in the kinks or bent portion. As you can see in the first picture, so under the fluid picture, so you can see that the unsaturated hydrocarbon tails has kinks or bent portion in their tails. So if all the fatty acids tails will have saturated bands, or there is no double band in between the hydrophobic tails, they will push against each other at low temperatures, making the membrane more viscous and rigid. So this is addressed by the appearance of kinks that push adjacent phospholipids to maintain fluidity at low temperatures. The membrane is very much like fluid because the molecules can move freely past one another, and a mosaic because of the diversity of proteins embedded in it. So many of these proteins molecules drift laterally like floating icebergs within the bilayer. So the number and kinds of proteins can vary in the plasma membrane and in the membrane of the various organelles. So it is important to understand the function of these membrane proteins, especially with regard to medicine, because most of the drugs bind to them. So for instance, the omeprazole or the Prilosec is a drug that relieves heartburn by blocking some of the transport proteins that pump hydrogen ions into the stomach cavity. So another example is the antidepressant drug which you call the fluoxetine or Prozac, so which prevents receptors on brain cell surfaces from reabsorbing a mood-altering biochemical called your serotonin. So there are factors that affect the fluidity of the cell membrane, which includes your temperature, the cholesterol, the saturated and unsaturated fatty acids. So as what I have mentioned earlier, that this temperature will affect how the phospholipids move and how close together they are found. So they are closer together when it's cold, and they move farther apart when it's hot. So when we say, when in terms of the cholesterol molecules are randomly distributed across the phospholipid bilayer, so helping the bilayer stay fluid in different environmental conditions. So the cholesterol holds the phospholipids together to separate too far, let unwanted substances in or compact too tightly, restricting movement across the membrane, as well as one of the factors that affect the fluidity of the cell membrane includes the saturation and the unsaturation of your fatty acids. So fatty acids are what make up the phospholipid tails. So the saturated fatty acids are chains of carbon atoms that only have one single bond between them. So as a result, the chains are straight and easy to pack tightly. And when unsaturated fats are chains of carbon atoms that have double bonds between some of the carbons. So the double bonds create kinks in the chains, making it harder for the chains to pack tightly. Now, each type of protein that is associated with the cell membrane has a specific function. So all of these membrane proteins, however, can be classified into two groups. So the two groups that are the two classifications of your proteins includes your peripheral proteins, which are those that are found in in only one side of the membrane. So they interact with the internal or external surface of the phospholipid bilayer. And by contrast, integral proteins are those that penetrate the hydrophobic core of the lipid bilayer. So they may or may not span the entire lipid bilayer. So if they do so, they become transmembrane proteins. So these proteins may be functionally classified as the channel, channel protein, carrier protein, adhesion protein, recognition protein, or receptor proteins. So transport proteins may either be channel proteins or carrier proteins. Now, channel proteins, as their name implies, serve as tubes or channels that allow passage of molecules across the membrane. So from the word channel itself, it implies the passageway of the proteins. So these transport proteins are open channels through which a substance moves on its own across a membrane without requiring any energy. So carrier proteins are involved in the transport of molecules, but they change shape as they receive certain substances. So this change in the shape allows the movement of the specific molecules across the membrane. So that's in protein. This protein is very specific to the substances it transports. So unlike the channels, it is not that specific when it compared to this carrier proteins, which is more specific to the substances it transports. So the next one is what you call the adhesion proteins. So these adhesion proteins in the plasma membrane fastens adjacent cells together in animal tissues. From the word adhesion or adhere, or it adheres one cell together from the other. Recognition proteins function as unique identity tags for each cell or species. So they also help in recognition when the body is being invaded by pathogens so that the necessary immune response may be triggered. Like for example, if a pathogen may invade or may enter inside the cell, the recognition proteins would release necessary antibodies or antigens to hinder the entrance or to kill or to stop the entering of that pathogenic microorganisms inside the cell, thus protecting the cell from that pathogen. So receptor proteins bind to specific extracellular substances such as hormones or toxins or to molecules on another cell's plasma membrane. So they have certain shapes that allow all the specific molecules to bind to them. So binding to these proteins triggers the change in the cell shape, which brings a cellular response that is related to metabolism or the movement, division, or even cell death. So different receptor proteins occur on different cells but are critical for homeostasis. So aside from the phospholipids and proteins, we also have carbohydrates that is present in the plasma membrane. So these carbohydrate chains are attached to either the phospholipids or the proteins. So as mentioned in the previous units, we have phospholipids with attached carbohydrate chains are called glycolipids. And proteins with carbohydrate chains are called glycoproteins. So the carbohydrate chains occur only on the outside surface of the lipid bilayer or the peripheral proteins that occur on one surface or the other. So in animal cells, the carbohydrate chains attached to the proteins give the cell a sugar coat with which you call the glycocalyx, so which imparts protection and other functions such as the cell-to-cell adhesion, reception of signaling molecules, and to cell-to-cell recognition. So this carbohydrate in the cell's exterior can vary in the number and sequence of sugar. So it depends in whether the chains are branched. So each cell in an individual has its own identity because of these chains. So this is the very reason why the transplanted tissues are often rejected by recipient individuals. So once immune system may be able to detect that the foreign tissue cells do not have the appropriate carbohydrate change to be recognized. Now, for example, if a person will undergo kidney transplantation, the doctors would check first the compatibility uh, of the donor to that of the patient. If the donor's kidney is not compatible with that of the patient, eventually the patient's body will reject the organ, since the immune system is will detect that the implanted kidney is has a foreign tissue cells that is not appropriate to the carbohydrate change that is present in the patient's body. So the main function of the plasma membrane is to regulate the passage of molecules into and out of the cell. So although simpler to hear, this is very important because the cell needs to maintain its normal composition while dealing with its changing environment. So one of the key aspects of homeostasis. So as we all know, it is selectively permeable, which means it only allows certain substances into the cell while keeping others out. So there is no required energy for molecules that can move freely across the membrane. So substances that are hydrophobic and with nature similar to the hydrophobic core of the lipid bilayer are able to diffuse across the membranes without using energy. So, however, the polar and relatively larger molecules, which are chemically incompatible with the hydrophobic center of the membrane, require energy to drive their transport. So, water, despite its polarity, relies on the small size of its molecules to cross the lipid bilayer. So, generally, small or non-charged molecules such as carbon dioxide, oxygen, glycerol, and alcohol can freely cross the membrane. So they are able to slip between the hydrophilic heads of the phospholipids and pass through the hydrophobic tails of the membrane. It's because these are also non-polar. So they have the same they are the same with the nonpolar region with these small and non-charged molecules. So the movement of these molecules depends on their concentration gradient. So what is the concentration? What is this? What we call the concentration gradient? So this concentration gradient, they move from. So what is this concentration gradient? This concentration gradient is the movement of molecule from a higher concentration to that of the lower concentration. So, for instance, a cell always uses oxygen to perform cellular respiration. So the internal consumption of oxygen in tissues results in low cellular concentration. So since the oxygen concentration always uses oxygen to perform cellular respiration, the internal consumption of oxygen in tissues results in low cellular concentration. Now, since the oxygen concentration is higher outside than inside the cell, the oxygen tends to move across the membrane into the cell. So on the other hand, the concentration of carbon dioxide is highest inside the cell because it is one of the byproducts of cellular respiration. So therefore, carbon dioxide tends to move from the inside to the outside of the cell. So here are the molecules that are impermeable and permeable to this membrane. So when there are the impermeable molecules includes your polar molecules or large molecules, which includes your glucose, amino acids, nucleic acids, and proteins. So for the permeable substances, it includes your lipids, alcohols, and small molecules such as your oxygen, nitrogen, carbon dioxide, and water. Now, To sum it up, the cell membrane is an organelle that is present in both plants and animals. So this organelle is selectively permeable as it only permits lethal substances to enter and exit the cell. So through this function, the cell eliminates waste products and foreign substances. It gains nutrients needed by the other organelles and regulates the required number of meetings. So the cell and the plasma membrane has three main structural components. The phospholipids, embedded in it are cholesterol, proteins, and carbohydrates. So phospholipids can move through rotation, lateral diffusion, and transversal diffusion. So lastly, the cell membrane is fluid due to the following factors. So temperature, cholesterol, and saturated and unsaturated fatty acid. So the cell plasma membrane structure is based on the fluid mosaic model proposed by Sir Jonathan Singer and Garth L. Nicholson in 1970.